Hi friends, I'm Anaga Srinivasan, first DC of MSc Biology. We already studied about the structure of sugars and now we are going to discuss about the different methods for representing sugars. Let's see. There are three different methods for representing sugars. The first one is Boyle-Stick model and the second one is Fisher's projection formula and the third one is Haworth perspective formula. Now let's see about Boyle-Stick model. Here, it displays both the three-dimensional position of atoms and bonds between them. In ball-and-stick model, the atoms or ball are represented by spheres and the bonds are represented by rods or stick. That is why it is called as ball and stick model. The double and triple bonds are usually represented by two or three curved rods. The angle between rods are same as angle between bond. In this, the sphere's color indicates chemical element of each atom. For the clear view of atoms and bonds, radius of the sphere is made to be smaller than rod length, which doesn't provide clear insight about space occupied by the model, and the sticks can be made flexible. Rather than ball and stick model, there are some more models, namely skeletal model, polyhedral model, composite model, and spherical model, which is also known as space filling model. But the ball and stick model is also an computer-based model. It can be virtual computer model which helps to illustrate shape, flexibility, and relative size. It is in true representation, hence the bond lengths are longer than radii of atoms. So accurate relative dimensions of molecules are not provided. It is an open model, so it is helpful for understanding geometry. In the figure, we can see the ball and stick model of glucose. The balls are in different colors, which shows the different chemical element of each atom. The next method is Fisher's projection formula. In Fisher's projection formula, the two-dimensional representation showing configuration of stereocenter. It is mainly used for the three-dimensional sugar structure on paper representation. In this, the horizontal lines are represented bonds projecting forward and the vertical lines represent the bonds projecting to the rear. In this method, the stereocenter is the only atom in plane of the carbon paper. It is flattened into two dimensions and it is commonly used for linear sugars. In the figure, it is an example of Fisher's projection formula D-glucose. Next method is Haworth perspective formula. It is proposed by W. N. Haworth and it shows cyclic structure representation of sugars in simple two-dimensional perspective. In Haworth perspective, the hexagon represented by pyranose and pentagon represented by furanose. It is a six-member ring is entitled to be make its plane almost perpendicular to that of the paper, with the bonds close to the reader, though thicker than those farther away. It is mainly useful for sugars which have a ring structure. There is an example of glucose of Haworth perspective formula, alpha D glucose and beta D glucose. There are some rules for conversion of Fisher's formula to Haworth formula. Let's see what are the rules. To convert the Fisher projection formula of any linear dihexols to a Haworth perspective formula showing the molecular cyclic structure, draw the six-membered ring having 5 carbons and 1 oxygen at the upper right. Next, number the carbons in a clockwise direction beginning with the anomeric carbon and then place the hydroxyl groups. If a hydroxyl group is to right in the Fisher's projection, it is placed pointing down, that is below the plane of the ring in the Haworth perspective. If a hydroxyl group is to left in the Fisher's projection, it is placed pointing upward. The terminal CH2OH group projects upward for the D enantiomer and downward for the L enantiomer. The hydroxyl on the anomeric carbon can point up or down. When the anomeric hydroxyl of a dihexose is on the same side of the ring as C6, the structure is by definition beta. When it is on the opposite side from C6, the structure is F. Here is the diagrammatic representation of conversion of Fisher's projection formula to a Haworth-Perspedt formula. Here we see how we can convert the glucose. We know that glucose is a 6-carbon atom and its formula is 6H12O6. In glucose, the hydroxyl group CHO having the double bond between CO is brought down and hence they possess partial charges. That is, delta negative for oxygen and delta positive for carbon. The carbon having slightly positive charge attracts with the oxygen in OH of fifth carbon of glucose and the oxygen having slightly negative charge attracts with the hydrogen having positive charge of OH of fifth carbon of glucose respectively. Then coil it into a cyclic structure having 5 carbon and 1 oxygen. Then place the hydroxyl group according to the rules for conventional formula. This is the structure of D-glucose in Fisher's projection formula and Haworth-Perspedt formula. Next example is mannose. Let's see how we can convert D-mannose to the Haworth-Perspedt formula. The pyranoses are 6-membered rings, so start with 6-membered Haworth structure with the oxygen atom at the top right. Number the carbon atoms clockwise starting with the aldose carbon. For mannose, place the hydroxyls on C2, C3, C4 above and below the ring respectively. Because in the Fisher projection they are on the left and right sides of the mannose backbone. Next is the other example, ribose. Let's see how we can convert D-galactose into alpha and beta L-galactose. For D-galactose, the hydroxyls are oriented below, above and above the ring for C2, C3 and C4 respectively. The hydroxyl at C1 can point either up or down. There are two possible configurations, alpha and beta, at this carbon. For the beta L-galactose, use the Fisher projection formula D-galactose to draw the correct Fisher representation of L-galactose, which is its mirror image. The hydroxyls at C2, C3, C4, C5 are on the left, right and left sides respectively. Now draw the Haworth-Perspedt, a 6-membered ring in which the OH groups on C2, C3 and C4 are oriented up, down and down respectively. Because in the Fisher representation they are on the left and right sides. Because it is the beta form, the OH on the anomeric carbon points down, same side as C5. Another is an example, fructose. These are what we referred, and thank you friends for listening my class.